Okay, our last presenter and funder is Paula Whiskey. She is from the Sloan Foundation, and people may already know that she's been very much involved in the microbiome of the built environment, so we're pleased to have Paula here.
Well, good afternoon. Delighted to be here. Now, I, unlike the three previous teams, need to say that the talk is my own and does not necessarily represent the views of the Alfred P. Sloan Foundation. I want to start by thanking the National Academies for undertaking this study. I want to thank the co-sponsors, the EPA, the CDC, and the VA, each of whom has put up a lot more money than the Sloan Foundation. I want to thank the committee for agreeing to serve. I can tell this is going to be lots of lively discussions. And I also want to thank all of the Sloan grantees, reviewers, advisors, and staff who have helped guide our work in this area. So, all right, so I'm going to do three things today. I'm going to tell you a little bit about the Sloan Foundation. I'm going to tell you a little bit about the microbiology of the built environment program. And then I'm going to tell you the story of how we ended up being a sponsor of this study, management of Legionella in water systems. Okay, so I don't know where I'm pointing this. All right, so who's the Sloan Foundation? We're a New York City-based philanthropic institution. That means I can see those cooling towers that are regulated by the Department of Health, a long-term partner of the Sloan Foundation. The Sloan Foundation supports research and education, generally in science, technology, economics. We have a strong interest in developing early career scientists, and we generally develop a program strategy and then invite proposals. We rely on advisory committees, peer review. We like to partner with federal agencies and industries, and we usually in our research programs, support a field or program for 10 years. Now, one of the things that we don't do is we don't fund health or biomedical research. So I'm delighted that all three agencies have been doing lots and lots of work in Legionella for a long time, but that's about how I got here. All right, so we started a program called the Microbiology of the Built Environment, started funding work in this area in the mid-2000s. It came out of our work in the bioterrorism program where we were trying to make buildings safer against biological threats. Let me step back for a moment. Anytime I say we, it's really the grantees. Okay, we're like a talent management agency. Anyway, so we were trying to make buildings safer against biological threats. And we started thinking those biological threats are the needle in the haystack. What's in the haystack? And around this time, there were all sorts of new culture-independent methods for detecting microbial profiles. So we convinced two prominent microbial ecologists, or they might call themselves something else, really extraordinary life scientists, to move indoors with their tools and techniques. Craig Venter, who did the New York City Air Project, which I'm not going to talk about today, and Norm Pace at the University of Colorado Boulder. Over the past, however many years, we've spent $50 million. We've funded approximately 160 grants. And we're really delighted because last summer, the academies came out with a consensus study on microbiomes of the built environment, a research agenda for indoor microbiology, human health, and buildings. And this was co-sponsored by NASA, NIEHS, and EPA. And again, it's an excellent summary of the Mobe program if you want to learn more and really start to understand how buildings are indoor ecosystems influenced by design, occupancy, and operation. I suggest you read it. And if you're a funder, I suggest you read it even more because there's lots of important directions. And again, since we don't fund health, the health implications of these studies, there's really a lot of opportunity. There's also a Gordon Research Conference system on the microbiology of the built environment, integrating human health with building microbiomes. Again, I can give you that later. All right, so now let's move on to this topic. How did I get here? Well, as I said, one of our first grantees was Professor Norman Pace at the University of Colorado Boulder. He is a very famous microbial ecologist, came up with the concept of the tree of life. So in 2005, we awarded him and his team a grant to begin cataloging the indoor microbial world. That was the purpose of the grant. Begin catalog. So Norm, take your tools and techniques. What do you want to study? Norm became very, very interested in drinking water and did lots and lots of studies. But the one that became very famous was the one published in 2009 in PNAS entitled Opportunistic Pathogens Enriched in Showerhead Biofilms. And it's so interesting that something else that the Sloan Foundation supports, Science Friday, even did a story on it. What's hiding in your showerhead? A new study says showerheads may harbor microbes that can be harmful to people with compromised immunity. Biologist Norman Pace describes what his lab found growing inside some showerheads and explains how your shower spray may help organisms spread. Now, what a surprise that I was one of the lucky building owners or apartment owners that had a showerhead in that study. I did not. So I thought about my risk assessment, but again, this really brought a lot of attention to people start thinking about drinking water is not sterile, microbes are coming out of my shower. I mean, it's a very exciting time. All right, so Norm really, Norm got a lot of people excited and put together in 2012, an American Academy of Microbiology workshop in Boulder, Colorado. And a number of people sitting in this room were there and, again, had a great time, lots of good discussion, and they came out with this report. You can't read this, okay, but I put it in because it's a box on the opportunistic pathogens in the water distribution system. I think they've all been mentioned, but anyways, there's things to do there. Now, I told you we did not fund work on health, but at this meeting, I met Amy Pruden. And I said, Amy, I really would like to work with you. We really don't have anyone working on premise plumbing. And she said, well, Paul, I really want to study these opportunistic plumbing pathogens. I'm like, well, as long as it's part of the ecosystem, that's fine. But like, tone it down because we don't fund health. We don't fund pathogens. We really want to understand the ecosystem. And Amy and the team at Virginia Tech, again, this time in environmental engineering. So think back, Norm is a biologist in life science. Now Amy's in an engineering department. Okay, and there were lots of, I just putting this publication up, which actually was published last year, but the results were shared before that time, that talked about water heater, temperature settings, water use frequency on the building plumbing microbiome. This is really interesting. So again, we're not talking about all the hard work that's going on at CDC or the EPA or at the VA. These again, Sloan Foundation is trying to elucidate the microbial ecology in the built environment where people spend 90% of their time. Seems like an interesting place to study. And so here's another paper. All right, so next comes Ruth Buerkle. Now, Ruth and I, back in the olden days, worked together fighting bioterrorism. And many times Ruth cautioned me on the Sloan Microbiology of the Built Environment program because she said, Paul, are you sure they're not just on a fishing expedition? And I said, well, Ruth, they are because we don't know what's there. We've got to do some fishing. Anyway, so Ruth approached me and started talking about Legionella. And I said, well, I think you should go talk to Amy. I think you should go talk to Norm. And next thing I know, we had this fabulous workshop at Emory, from watersheds to showerheads, a workshop on Legionella research and policy. And you'll see at least 3 of the 4 people in this particular picture were at that workshop. I think there are a lot of other people in the room who were at the workshop or presented the workshop. It was a very multidisciplinary workshop. There were, again, I think there were people from all the different agencies that have presented today. There were other people from water companies, people who are selling different things. It was great. But the big takeaway for me from this meeting is that generally we have safe drinking water, but did we have safe breathing water? And now let's go back to Norm. After he did his determinations, he stopped taking showers. So anyway, we were fortunate enough at that meeting that Laura Ehlers came to it. And so that's how I got here today, because I was so thrilled that all the different parts of the Sloan Foundation program were coming together, and actually we could fund something that had something to do with health. So I'll stop here. Happy to answer any questions. Thank you. Okay. Okay.
Mark Lachevalier. So that's great. And this, if I count that, you're probably past the 10-year mark on this.
Yes.
So what's the future? Is this the end?
Right. So
is this the final swan song, or?
No, I'm still going to be spending a little money this year and next to try to engage other stakeholders so that they'll put up the big bucks to do all the health studies that we did not do. And again, that's why I showed the Academy's report. But a few people in the room know I trained in chemistry, and so I actually have a new program on the chemistry of indoor environments to elucidate the indoor sources, the chemical and physical transformations, and the role of the occupants and the role of building in indoor chemistry. And it's really exciting. And again, same type of strategy. You take the people already working outdoors with their tools and techniques and move them indoors.
So you think some of that chemistry could be impacting the microbiome, and we could?
Oh, yes, and there will be some people who have been studying, who are in the Mobe program, who are going to move to the chemistry program, because microbes are important building occupants. So now is it break time, Joan?
Well, not yet.
Okay.
I'm not going to go to break yet. I did want to ask you, in the 10 years, what's your feeling of how the tools have changed? From 454, I mean, there's a lot of different instruments, whole genome, we heard about whole genome sequencing from CDC, and all right. So what's your sense of that change and how it's affected the ability to assess what's going on in the environment?
All right. So one of the biggest things that happened is it became cheaper and cheaper and faster to do sequencing. So that was great for the program because we could fund more studies and so on. In some cases, it became more difficult to do the analysis because you had so much information. So we spent money, some money in that area. I think now, at least from the research perspective, that people have the tools to both do whatever type of sequencing, whether it's, targeted or whole genome or something else, some other, and to do that. But I don't think that those tools have translated well to, say, environmental testing laboratories.
All right, I'd like to ask the audience if they have any other questions for our previous presenters. If there's anything else that you didn't get time to discuss. Committee members? All right, check.
I'll put Eric back on the spot again. No.
Eric here.
I mean, is there, did he leave? He's left? Is there a process or a background or any precedent for something that's been controlled via a treatment technique to change over to be controlled by an MCL?
So Chuck, let me make sure I understand this. So has EPA ever promulgated a treatment technique for contaminant and then subsequently revised the regulation to promulgate an MCO? I'm not aware of any such transformation. So what would have to take place legally, I believe, is to do a treatment technique, the agency has to find that it is not technologically or economically feasible to ascertain the level of the contaminant. So I guess legally it would be possible if science changed between promulgating a treatment technique and then subsequently revising it if we determine that it is technologically and economically feasible to measure the level of the contaminant. So I guess it's possible there, but there is no precedent that I am aware of.
All right.
And
Now,
I'll turn it back over to Paula for all other questions.
We've got a comment here, and then how many public speakers do we have, Laura? 4 or 5 on the list? Okay. Let's go to the online question, then Michelle.
Yeah. Oh, there, he's gone. It's a question for you, Eric, even if you're in the back. With the Flint situation, is EPA reconsidering red water control in terms of health impacts and Legionella? Is that on your board? Is it on your list of ongoing projects? Because it actually, taking care of the large buildings is one thing, but regulating corrosion in systems is in your realm, so. Sorry.
Paula, would you like to answer this question?
I am a special government employee of the EPA for about 3 days a year, but not on this topic.
So, all right, Michelle, let me, when you say red water, I assume you mean corrosion, iron-based corrosion events. All right, so we're talking about a different regulatory requirement than the ones that I've mentioned previously. We're talking about the lead and copper rule right now. And so there is a regulation, the lead and copper rule, that does require corrosion control, which, while not targeted at preventing red water, does require for all systems serving 50,000 or more people, or for small systems that have elevated levels of lead and copper to institute corrosion control. So yes, we are in the midst of evaluating revisions to that rule, but not for the purposes that you are suggesting. We are focused in the context of that rule on minimizing lead and copper exposure.
Okay, we've got an online question.
We have 2 questions from Marian Heyman, who is at the Connecticut Department of Public Health. She has one for Brian and one for Laura. To Brian, she asks, should we be getting environmental isolates from sporadic and outbreak cases to you for further genetic study to include, for instance, in a national database? In Connecticut, all environmental samples are performed by private environmental microlabs. Would you want us to approach these labs about sharing isolates?
Hi, this is Brian Raphael CDC. So I think there was 2 parts in that question. There were the cases of sporadic disease. I can say that we are working with several states that have sequencing capacity either at local or state level laboratories where we can share sequences. In many cases, a state agency, a state public health agency, can send isolates to us for reference testing, and our intention is to sequence any clinical isolates from sporadic or disease associated with clusters or outbreaks. The other part of the question has to do with environmental isolates, and I would say that we certainly can work and collaborate with various jurisdictions when it comes to environmental isolates from routine testing. There are so many isolates that we really need to work with jurisdictions to find out what the best scheme would be to sequence strains that might be useful for ecology studies. However, when it comes to environmental isolates that are collected as part of investigations of Legionnaires disease clusters or outbreaks, we can certainly work with any agency that's involved with those investigations to sequence environmental isolates as part of an investigation.
And we have a second question again from Marian Heyman at the Connecticut Department of Public Health for Laura. Marian writes, "We all try to emphasize the importance of getting cultures on patients. However, we are frequently told by LTCF and others that they can't get sputum specimens on patients. Can we take a lesson from the tuberculosis folks who can get sputums from anyone? Maybe you or your colleagues can create some teaching videos to instruct staff on how to get sputum samples on tough patients. Our tuberculosis folks say they have several strategies and tricks that really work. This could be included in a packet with some of your other materials."
Hey, Marian, this is Laura. Thank you for that. You always have great thoughts and input. I think that's a great suggestion. I mean, it is interesting. We work with several labs and health departments where, amazingly enough, they get lots of respiratory cultures. And the trick with respiratory culture or respiratory specimens for Legionella, it doesn't have to be a perfect specimen. I know that a lot of labs will throw out specimens because they have too many polymorphonuclear cells or, you know, too much contamination. The fact of the matter is, and Brian has taught me this, that it really doesn't have to be the greatest specimen ever to be able to isolate Legionella just because of the process involved. So I think that's a great point. I guess I really hadn't thought about us doing like a video, but we're up for it. We'll be calling you to help us with that.